start sleep, okay? I'm going to turn off my...
As you all know, we are here.
Okay.
We are here.
Okay.
Thank you.
Tangriani for the introduction, and welcome to my associates here in Raleigh and to all of my colleagues in Brazil. As you see, the is focused on soil biological quality and its somewhat contribution to sustainable agriculture. The picture was actually taken in Porto Alegre from a long-term no-till soil, and you see the high organic matter at the surface and the mineral matter contained within with a very good structure. He's going to be okay. Okay. Manage our lands, and we manage the lands that we try to get productivity. But as you can see, we often fail to get productivity. We get degradation of our land. So it's intense to produce food and fiber, but sometimes we treat our most valuable resource much like dirt rather than like soil. There's been a movement to define soil quality, because soil quality is a process of understanding how soil functions. And soil quality has been determined by various properties and processes of soil, and that's broken down into chemical, physical, and biological components. We can measure total organic carbon, and some decade or even 2 decades of research on this area, most total organic carbon is probably one of the best indicators of soil quality, because it incorporates not only chemical but it also influences physical and biological properties as well. But you can see that we can measure a number of chemical indicators: total nitrogen, pH, and a number of typical soil testing values, such as inorganic nitrogen or phosphorus or potassium. And the electrical conductivity can be important for salt accumulation. Capacity is influenced by mineralogy, but also by the organic matter. And with products, we can actually detect that and determine whether the soil chemistry has been changed or not. List of soil physical indicators. Many of them some are inherent, such as humidity and texture. These are inherent characteristics. We will not change them through management, but other characteristics such as porosity and water infiltration, aggregate distribution, these things can be changed by management, but they often take a very long time to actually biology interact with the physics so that they actually change. So it takes years for some of these things to have a noticeable change. Biological indicators are things that we know about: microbial biomass, carbon and nitrogen, soil respiration, mineralizable nitrogen, and a suite of enzyme activities. There are many different enzyme activities that can be measured, and this is part of the problem, or the conundrum that we have, is that we have many different indicators, and the biological properties tend to be very temporally sensitive. So it can change dramatically from day to day or season to season, and certainly from year to year. But the fact that they change so dramatically in a period of time gives some limitation. And so we need indicators that are robust over time, and that reflect management conditions, consistent management conditions, and don't change too rapidly so that they cause unknowable variation. Investigated in the soil quality arena, and this is one of the reasons why the Natural Resource Conservation Service of USDA dug into the concept of soil health. Health being a factor that is a living factor, and so the emphasis on health, on the biological component, is the reason why there's this new initiative on soil health. And this initiative has just started in the last few years. It incorporates many of the concepts of the soil quality paradigm, but it focuses more on the biological activity or the soil health itself. We're on understanding the influence of management and organic matter content on nutrient supply. This is complicated, and it's also a little bit outdated because it was part of my PhD work. But this top left panel here shows the influence of conventional tillage here in these two lines here, row tillage on the organic carbon supply, of cropping intensity. So the cropping intensity would have been 0.5 under continuous wheat, and then a rotated wheat soybean, and the largest, the highest value under continuous wheat soybean. So it incorporates that, but it also incorporates the idea of adding fertilizer or not, nitrogen fertilizer in grams nitrogen per meter squared. Under conventional tillage, the levels were low. Under conventional tillage, there was no effect of fertilizer nor of cropping intensity, but under no tillage, there was of fertilizer and cropping intensity on the soil organic carbon level. So the more intense the system, the more nitrogen, also the more soil organic carbon. That was also reflected in microbial biomass carbon, so similar trends, but a little bit more dramatic. So the biological component is a little bit more expressive than this total pool of carbon in total organic carbon only. We can have a ratio of the soil organic carbon as microbial biomass carbon. We have measures of mineralizable nitrogen and mineralizable nitrogen here, mineralizable carbon, microbial biomass carbon. So microbial components tended to be a little bit more responsive, and this led me to my interest in the microbial aspect after. It's very important to understand that this talk is going to be about nitrogen, but it's also going to be about carbon. And a strong relationship between nitrogen and carbon. In fact, in the red line here, we have an input of mineralizable carbon through crop residues or roots. We decrease in microbial activity at some point, and then the substrates tend to decline, and then we have a declining microbial activity. It's steeper than the initial level, but it tends to decline with time. The nitrogen in soil, as inorganic nitrogen, tends to dip in the initial reactions because the microbes growing intensely have to utilize that nitrogen that was in the soil, and so there's a net immobilization that can occur. At this time, there's a net mineralization. And there are differences, and there's quite a large literature on carbon to nitrogen ratio, and the carbon to nitrogen ratio does affect mineralization in the short term, but in the long term, almost all residues that are added to the soil in a form of carbon-rich residues, they all lead to net mineralization at some point in time. It's just a matter of time when they mineralize, and that's the understanding that we have to come to, is when does the net immobilization convert to net nitrogen mineralization? Microbial activity sequesters nitrogen into organic matter. These were taken from my PhD study as well, and I was interested in this strong effect of no tillage versus conventional tillage. This was a long-term study. This data were taken after 8 years of the comparison between no tillage and conventional tillage. And that with carbon mineralization, there was this consistency that occurred during the incubation. So the x-axis is the days of incubation, the y the potential carbon mineralization, the actual CO2 release during incubation. And we saw very consistently, independent of crop, that's between no tillage and conventional tillage. No tillage always leading to greater carbon evolution. Greater carbon evolution was because of the potential that was stored up in the soil over those 8 years. Carbon actually being emitted more with the conventional tillage than the no tillage, because that's why there was less potential under conventional tillage, because it had exhausted its carbon that being put in through the crop residues, but the no tillage was retaining that. And so the consistency among crops, in the nitrogen response, the potential nitrogen mineralization, there was also very strong consistency. These led me to the idea to pursue, well, I was interested in the long-term carbon mineralization to be able to predict nitrogen mineralization. And at the data further, we selected 5 different soils. There was a soil with low carbon mineralization, then increasing with time, and with the samples. This happened to be a sample with high carbon mineralization. The end really into defining what was microbial biomass, because we knew that the microbial biomass is the component that is important in this case. The carbon mineralization into the actual weight, if we take the derivative, we saw that the rates of the carbon mineralization were also consistent. There was no crossover between any of the lines. Also is that we were more interested in this steady state rate of carbon mineralization. Actually determine this steady state mineralization. And this was a revelation that came. Earlier work had shown that yes, there's a flush of CO2 when we have actual dry wetting of the soil, but this was a clear indication that actually maybe we could just use a very short-term assay to be able to predict a longer-term assay. And this of even for 24 days is very short-term, because many of the previous studies looked at 30 weeks or 32 weeks or 26 weeks. There was a long-term aspect to it. So by studying my PhD in Texas, moving to a postdoc in Alberta, and then my first job in Georgia, measuring similar things because the interest was in organic matter, and we saw that there was a pretty stable relationship. Now, some of these aren't ideal as such, but I asked a colleague that was in ARS at the time, who was in Maine, to be able to fill out this quadrant where this area is cold and dry, Texas is hot and dry, Georgia is warm, moist, and Maine being cold and moist. So we had 4 different extremes. And this colleague in Maine was Wayne Honeycutt, who is now the technical director for the Science and Technology Policy Division, USDA, NCS, and who was really responsible for the soil health movement being implemented by NRCS. That we were looking at in 3 days, and it's a technical technicality of why we use 3 days. It just happens to be a convenient measure to do it in 3 days. You can start the incubation on Friday and take them out on Monday, and it will be a 3-day incubation. It will cumulative carbon mineralization in 24 days, and relates strongly to the soil microbial biomass carbon. So is the flush of CO2? It's probably an indicator of microbial activity, but it's also an indicator of microbial biomass. So something in between, isn't it? And to blow up this bottom graph a little bit larger, we can see that there is this strong relationship up to a point. And these data here are not necessarily unique, but they're also not typical, because we don't always see this flattening out of this relationship. But this flattening out is likely due to nitrogen immobilization that is occurring in some samples. And so we have to be aware of that. And that's something that our research will to out more in the future. So the procedure of determining the flush of CO2, and there are certain technical steps that I think it's important to follow a protocol entirety, otherwise the results may be somewhat skewed, and so we try to be as consistent as possible. So far is getting a representative sample in the field. This is very important. I've had Leonardo Dace accompany me with me in the field, and we try to get at least 8 cores in a field, and we then dry the samples. This is actually unique for microbial analyses. Most microbial ecologists would not dry the sample, but we've documented that in the past we can dry the soil, we can pre-incubate them a little bit and then get even determined microbial biomass. But the idea here is that we're actually determining the flush of CO2 from a dried sample that is re-wetted. So it's important to dry the sample fully as possible. And we do choose a soil temperature or a temperature to dry them at to standardize the procedure. So the section procedure is important. It's important to represent the soil not only spatially across the field, but also with depth. And so we are very careful in determining the exact depth. In many of our studies right now, we're taking 0 to 10 centimeters, 10 to 20, and 20 to 30 centimeters. And be represented by these 3 bags here where we put them in paper bags because we're intending to dry the samples, and so we don't want to put them in plastic bags. 3 days. It can go longer. The longer it dries is not a concern, although it's possible that if it were dried for more than, say, a few months, that continuously dried with high temperature, possibly some negative reactions could take place. The next step is to homogenize the soil, and this is an important part also because also is to not to disturb the soil dramatically, but we do need to homogenize the soil. So the fermentation process is to then lay out the soil and to be able to pick out any large pieces of residue, such as this piece of soybean stubble here. Fine matter where it's been pounded with a pestle and with a mortar and mortar and pestle, and so then it becomes small enough so that it can become homogenized, but not too small that it's grinding to a powder. And that is then to pass it through a sieve. We pass it through a 4.75 millimeter sieve. The soil that passes through under now, and then all these pods that still remain, and so they have to be pounded through. And at the end of this process, we're left with only the stones that are greater than 4.75 millimeters. So this produces a product that is relatively homogeneous. It's at least relative because we want to retain some of that structure. We want to have those aggregates that have micropores within them. We want to have them so that they have macropores as well. So it retains some of the inherent characteristics, but it compromise between retaining an intact core and having a homogeneous soil sample to work with in the lab. To wet the back up into these little bottles that we use, we have graduated bottles. They accommodate up to 60 grams or 60 milliliters of material. And we add the soil there, and we 50% water-filled pore space. So we have to calculate what the volume of the soil is in these little bottles, and then calculate how much water to add to get to 50%. Because we don't want to oversaturate it, but we want to give it enough water. So these things that we have in this setup. We have a 1 liter canning jar with a lid, so that's displayed here. We have 2 graduated bottles with 50 grams of soil, left and right of the sample here, and water-filled pore space. Then we have here in white here. This is a plastic vial that has a screw cap, sealed afterwards to hold 10 milliliters of 1 molar soil hydroxide to absorb the CO2, because CO2, being a weak acid, will be absorbed into the alkali, the strong alkali of the solution, and then we can determine how much CO2 went in there. So it's part of the respiration of the soil. And then we also have another vial just to have a little bit of water in there to maintain humidity, so that we don't actually dry the soil out more than, because we do open them several times at 3 days and 10 days to replenish this trap. So 1 bottle of soil is used for pre-incubation for 10 days prior to chloroform fumigation to estimate soil microbial biomass carbon. And the other one goes for 24 days to determine the cumulative carbon and nitrogen mineralization. Molly is adding the water to the samples. Ellen is filling these vials with alkaline. The dispenser here, and she's putting it into these jars next to her on the lab bench. Here's actually taking the traps, the alkali traps out of the sample and filling it with this dispenser here, putting a new trap in there, and you see the same thing here where Tangri and LCU are work soils. They have the two soils in there. There's the water container and this vial of alkali. These are the sealed containers where they are, the alkali will then be titrated afterwards. Will be determining how much CO2 was actually evolved from the soil and in this sample here. And then samples are put back into the box and into the incubator to incubate at 25 degrees Celsius. Titrating the absorbed CO2 in the alkali with hydrochloric acid. This is shown here with this color indicator. We actually precipitate the CO2 with barium chloride. Barium chloride is strongly absorbed to the carbonate that's present in the sample, and so it precipitates out. It actually takes the CO2 away from the solution and puts it into a solid. So the solid is actually just stirred up and mixed. So it's away from the acid that we're adding through this titration step. And the indicator is then our endpoint. When it turns from pink to clear, then we know that we've reached our endpoint. And the process is accommodated by a blank sample where we have no exposure to the soil. So we know how to calculate the blank from there. Of the quantity of CO2, where we have the milliliters of acid of the sample, 2 liters of acid of the blank, and that difference, that quantity there is multiplied times the normality of the acid, which is typically 1 in our lab. It can be around 1. We have to actually determine what that milliequivalents per milliliter is. And then a milliequivalent and mass conversion ratio, and this is from the milliequivalents of the acid required to neutralize the barium carbonate, remaining alkali. It's actually, there are 2 milliequivalents associated with that, and then milligrams of per of CO2, and so we have that there, plus the conversion from grams to kilograms in there, so that's why the value is 6,000. And then we divide that by the soil weight, and typically we're using 100 grams in our lab. That's 2 soils of 50 grams each, and this would be for the 3-day analysis only. Because for 10 days, it would be the same, but then for 24 days or the 21 days, we're using a different soil weight because we've separated the bottles. We incubate it further for 24 days, and then the samples are sieved to homogenize them again, because we don't actually extract all of the soil. We only extract 10 grams of the soil. We put the soil, we shake it with 2 molar KCl. We actually the inorganic nitrogen and filter it through and collect the filtrate here, and runs it on the flow analyzer, where we have our samples now here in the background that are either frozen or refrigerated, and then the flow analyzer to determine ammonium and nitrate. In the protocol that can be looked at, and we've looked at some of these, and I'm here because this is the carbon mineralization in 3 days on the x-axis versus in 1 day. Rick Haney at Texas was a grad student with me during my PhD at University. He's now USDA ARS scientist in Texas. He prefers to use 1 day, and I'm going to explain a little bit later why I prefer still 3 days. But the relationship, so even in 1 day, as short as 1 day, and perhaps as short as we think that as short as 6 hours, there could be some good relationship. But the shorter we go, the more variations we're going to have, and the less sensitive we're going to be. But it does show clearly that even 1 day is possible to get a strong relationship. So extent of sieving, and this is a graph pulled from the paper itself, but I'm going to show you that so this shows the carbon mineralization in 3 days versus microbial mass carbon of basal soil respiration, which is essentially the rate from 10 to 24 days. The carbon mineralization during 24 days, the cumulative carbon mineralization, and looked at these treatments of moist intact soil, where dried, but the core was left intact. And then we dried it and then sieved it through 7.9 millimeters, through 7 millimeters, through 2 millimeters, or through 0.5 millimeters. So we were trying to look at the level of disturbance. So the ratio changes between the carbon minerals in 24 days, which would be, in this case, the top graph here. These are the rates that are depicted in the graph. You see that this rate here is a ratio of 6 times. There's times greater cumulative carbon mineralization in 24 days as there is carbon mineralization in 3 days. But the ratio changes with greater disturbance. So when we dry the soil, we have a lower ratio. Flush of activity. So the Cmin3 is actually changing. That's what's changing. The carbon mineralization, cumulative carbon mineralization, isn't changing much, because even this top point, you can see that it increases some, but not dramatically. It's really the carbon mineralization in 3 days that's changing. So the rates tend to converge. And so the width chosen is 4.75 millimeter at this level here. And it seems to correspond well with any of the drying treatments. Although drying really fine sieving, we do have some adjustment to make. This is where there's a little bit more of a problem. With microbial biomass carbon, there's also this relationship where with increasing that occurs, we have a lower ratio. The ratios that are from 2 to 8 millimeters here are very similar, so they're the same. And this 0.5 millimeter again is different. The nitrogen mineralization again, and in general it followed the same tendency, but of course we had a negative value here. That can be a part of the immobilization that occurs, and so that's the complexity of nitrogen mineralization that is there, is that it's net nitrogen mineralization, taking into account the gross mineralization as well as the mineralization that can occur. But you know, it followed the same pattern. The rate was highest with moist intact, and tending to converge into all of these dried samples, and even lower with the 0.5 millimeters. So we prefer not to pulverize the soil. One can pulverize the soil and still determine the flush of CO2. It's just that a different factor is going to have to be used. Water-filled pore space, and so the question is, why do we use that level? And the data we've collected in the past shows that in the soil type, whether it's low clay concentration or high clay concentration, all values that we tended to see were that there was a convergence around 50% water-filled pore space. And there are significant variations. These are individual values, so this doesn't take into account the averaging that could smooth out the data, but it shows that clearly there's a convergence around 50%, and we can go maybe as high as 60%. It could be a possibility without too much influence on the results. We also did a dust control experiment because some labs were inquiring. There was a telephone call that I had taken about in whether in commercial laboratories where drying the soil samples and they're out on the bench, they wanted to maybe mist the soils a little bit. And so what the data would show would be that up to 3 to 5% or so, one could say that the values are similar to if they were fully dry. But once they drop below with more moisture than that, then we're going to see a decline in the flush of CO2, and it's going to act more like a field moist sample. And the sample is not really what we're after. We want to see this flush of CO2. This is an expression of the microbial biomass and its activity at the same time. So the point that actually the procedure that we've developed is actually now commercialized with the Solvita soil test. It's a soil CO2 burst test. It has a setup where the soil is put into a glass container. In case they don't titrate. They use this color-coded paddle that's in there. CO2 is being absorbed. They also use water that they put into the jar and they pull into this plastic container and allow it to seep up by itself. So it's a new soil test, and certainly it is. It reveals the soil's underlying biological and sustainable yield, and nutrient inputs and improved yield sustainability. So this was a collaboration between my colleague Rick Haney with ARS in Texas and Will Brinton from the company Solida. Background in soil respiration and in compost maturation, and so he's got a strong scientific background. There are some interesting limitations, I believe, with this system. I say there are limitations at this point until the documentation is out there. And the issues are that they use only 1 day, and the ratio of 1 day of 3 to 1 day is about roughly 2.5 times that of 1 day. So we give our estimate with the 3 day. The other is that they use only 40 grams of soil, and we're typically using 100 grams. So again, there's another 2.5 factor in there. So you can see that already our method of using the standard procedure we use in the lab is probably more robust because we're using more soil and we're using over time period. The third is that they're actually saturating the soil from the bottom, and actually they reach near saturation in some cases, where it be 100% saturation. And this has a limitation on saturation, because if we actually exceed 70 to 80%, we're getting a decline in the flush of CO2. And this are from 3 days. The point being that there's significant research that has to be documented to show that there's a good enough relationship using this commercial test. And that's my only point. I think there's real value, but it needs documentation. Further now into characterizing some of the management. So this case here, we looked at cattle stocking rate in a grazing study on Bermuda grass and soil organic carbon accumulation. With grazing, we had a level of about 39 tons per hectare of carbon. With moderate grazing, we could increase the total of carbon in the soil, and with higher grazing pressure, it was higher than no grazing as well, but it was lower than with moderate. And we also had a hay value. So as an indicator of total carbon, and these are the data from the flush of CO2. We saw from the 0 to 2 centimeter depth, there was this also this increase with cattle stocking rate, and then tend to be a decline with the hay system. At 2 centimeters, the effect was subdued. There was this little bit of rise with grazing pressure compared to none, and then with the 4 to 6 centimeter, of course, these are very shallow depths, but it's the importance of the carbon itself with the accumulation. And in a permanent pasture, the carbon accumulation is typically at the surface. So to show that there's certainly a yearly response because these data were taken for the first 5 years, and this is carbon accumulation, the flush of CO2 that occurred each year. Also a strong response to grazing and strong depth effect. And they're all important things to relate to management. Another showed the effect of grazing cover crops compared to grazing of cover crops. So we have grazed and ungrazed cover crops under conventional tillage, again with different soil depths. You see that the flush of CO2 was greatest at the surface and being with depth. We did not of the grazing versus the ungrazed cover crops on the flush of CO2. And in fact, we didn't see that in any of the soil properties, whether it was total organic carbon or particular organic carbon, actual effect on microbial biomass carbon in one case where there was a greater microbial biomass carbon than ungrazed with microbial biomass carbon. Under the depth effect becomes even stronger, so the depth effect here at the very surface, there's a greater flush of CO2 compared to the conventional tillage. And then a strong decline with depth. In the effect that the no tillage is greater than conventional tillage at the 0 to 3 centimeters, no at 3 to 6 centimeters, and then a reversal that occurs because the conventional tillage is the residues, surface residues, incorporated into the soil, enrich these levels of CO2 with depth. there's no further input with the no-tillage. It's relying on root inputs only. But over the 0 to 30 centimeters, there's no between any of the systems. There was no response to grazing and a strong depth effect. Again, very strong reflection of the flush of CO2 on management. So mineralization, and essentially I'm just putting them up for convenience very fast, that the trends were the same. That the no-tillage greater than conventional tillage, in this case it happened to be at the 0, 3, and 3 to 6 centimeters, then no difference here at 6 to 12 centimeters, and then so with other lower depths. But at 0 to 30 centimeters, no difference between the systems. Consistency of responses between the flush of CO2 and net nitrogen mineralization. So reflect the nitrogen mineralization, and it also, the flush of CO2 also reflects the soil microbial biomass carbon. To show that this was the sampling the flush of CO2 on the x-axis versus nitrogen mineralization during 24 days on the y-axis. In the first longer-term study, this was the relationship we got. During the year of management, we saw the same exact configuration. We had different points along there, but we had the same slope. And in actually in this case, the 5th year of management, that had a similar response, but we had a few points that tended to be a little bit lower in net nitrogen mineralization, again, which may have reflected an influence of immobilization. So this net immobilization is a concern, and it's something that we have to be aware of. Changing of the relationship in different years of sampling, so it seems to be that the flush of CO2 is relatively stable, and certainly it seems to be reliable because we can use different years of the management and still that brought a point to my mind of what about seasonal distribution? Distribution should be reflecting in biological activity. In this here, this is a study from the PhD work as well, where we had no-till crops, continuous wheat, rotated wheat, soybean, and continuous wheat, soybean. The bottom graphs are the no-tillage. The top ones are the conventional tillage. You see that the conventional tillage was lower than the no-tillage at the surface, right? The surface was higher here. And the different points here are the sampling periods, which was planting, mid-flowering of the wheat, and then near harvest of the wheat. And so we saw this peak in almost all cases during flowering that occurs because the rooting itself of the crop is the highest potential then. We looked at the carbon dioxide flush, or the flush of CO2 in 3 days. We saw in the black point that this is conventional tillage, and then of the open circles, squares of tillage, we saw the strong tillage effect that was consistent. See this trend for a high value in mid-season compared to the at planting, and then tending to decline again with time at harvest. So it reflects what we measured under more cold conditions of the laboratory and of field sampling, not in repeated sampling throughout the year. So there is a seasonal distribution. It's influenced by recent carbon inputs via rooting, and there is this discernible effect even under these frequent samplings that occur. And there is variation, but in general, I would say that it's moderate random variation that's occurring, something that can be teased out, and we can see the seasonal pattern. So, as a measure of bioavailable nitrogen, we have this strong flush relationship between the flush of CO2 and net nitrogen mineralization, as well as microbial biomass carbon. So this has been documented in the past, and it's been documented in this study in the North Carolina Piedmont. Well, documented in the Georgia Piedmont as well. Same type of relationship where there's a strong relationship between the flush of CO2 and net nitrogen mineralization. And with the soils that we collected from different regions, there is this strong relationship between flush of CO2 and net nitrogen mineralization. So this the strong outlook for using the flush of CO2 as a soil test as a measure of bioavailable nitrogen. By Molly Pershing in a master's program, where she's actually growing plants in soils from different fields under different depth conditions, under different management, and documenting what happens to the plant growth. We documented earlier what happens to net nitrogen mineralization under highly controlled conditions, but these semi-controlled conditions in the greenhouse were documenting plant nitrogen uptake, first through dry matter production and then through the nitrogen analysis, but the nitrogen uptake will follow that of the dry matter uptake. Because all of the soils will be starved by the time they are harvested. You see plants are not in vigorous condition. They've utilized all the nutrients they could in 8 weeks and our expression of the biologically available nitrogen through this test. So that occurred. We have the flush of CO2 on the x-axis and accumulation, which is milligrams of dry matter per gram of soil. Because those were of different density, some are organic soils, some are soils. We used the volume of the container that we were using as the amount of soil. So we had to correct for the amount of soil. So in this sandy loam soil, that there's a greater flush of CO2 with 0 to 4 inches or 0 to 10 centimeters of and 20 to 30 centimeters. This effect is strong. If soil that is from clay loam location, this happens to be in the western part of North Carolina, the relationship occurred with depth, and seems to be maybe some textural effect, especially consider that there's another soil with a loam texture, and there was this depth, but then the textural effects. So one could say, well, maybe there is a textural effect that's occurring. But as we put those three sites together in gray, and now they become the gray dots here in this graph. And then we have another soil, which is from a sandy loam soil on the coastal plain, where there's no amendment. Then there was a history of low rate of poultry litter. So the flush of CO2 has increased because of the organic amendment. And the high rate of poultry litter, we have another relationship. And this is going to be explained by Molly in next week. That this point here is actually perhaps more due to the fact that we had a relatively low flush of CO2, but high dry matter production because of a significant amount of nitrate in the soil present prior to our intubation. And so that's because of the high rate of organic amendment, it led to high rate of organic nitrogen in the soil. As you notice, I went back here. The flush of CO2 went up to 200. I've expanded the graph now to 400. You're adding another soil, which is tends to be an organic type of soil here on the Tidewater region of eastern North Carolina, where we actually had a hayfield that was sampled, as well as no tillage in blue and conventional tillage in red. And they're also having some relationship and maybe some different slopes that are occurring. And so those different slopes are what we're interested in. Let's put them together as with soils from Virginia, Piedmont, and other pastures. We're seeing there's a really strong relationship for all these values that are in gray. We have an R square of 0.88. This is quite a very strong relationship that are actually different in nature. And these soils are ranging from anywhere from a sandy loam to a clay loam. And so we have different textures. We have different soil orders even. But most tend to be altisols. So there is this one inceptisol. And whether they're organic or mineral based. And so we're seeing this strong relationship independent of the soil type, which is encouraging. And our further step is to actually test this out in the field. Collaborators in Virginia that have been really willing to help, and we're looking at corn silage. These strips of corn silage, this is one strip of N rate, and this is another strip of N rate, and a third strip of N rate. We measured in the field, so we went in and hand harvested the silage. You might wonder, well, the look similar. This one tends to be a little bit more dried down than this one here. This actually, on the left, is the high nitrogen rate, and this is the low nitrogen rate. These here are perhaps 5 feet, 9 inches, let's see, so that'd be approximately 160 to 170 centimeters. And this is corn without side dress nitrogen. The farmer astonished that he could be producing that much corn without side dress nitrogen. We've all got corn grain production that's occurring in North Carolina and Virginia, so we're actually harvesting the grain as well. In wheat, we've had 1 year that Thais has been able to help us with wheat production, and planning to continue a few more studies on farm trials under long-term no-tillage situations with producers to look at wheat. And we're also looking at tall fescue. Smriti is looking at this to look at the flush of CO2 in tall fescue in both North Carolina and Virginia. And some of the locations have somewhat more than 20 different locations in North Carolina, throughout North Carolina, and a few locations in Virginia. Preliminary results. These are actually not of the means. These are from 3 sites with 4 locations, so all the data are here. But what we see is this flush of CO2 compared with the relative yield without nitrogen fertilizer compared with the highest rate of nitrogen fertilizer. So the relative yield scale, it would be 0 if there was no yield with no fertilizer during the season, versus 1, which would be the yield of without fertilizer compared to with fertilizer. And I expect that there would be some relationship here. So we had values that were increasing with increasing flush of CO2, but then we had this decline. And so this concern again with net immobilization that might be occurring. But again, like I said, these are the 4 points from the 4 reps. And so value would not necessarily be that much. It would be an average of more in this zone over here. So as so concerned about that, because we only have 3 sites. We're going to have more than 20 sites this coming year, and so we'll be able to document that further. In the same setup, the flush of CO2 compared to the relative yields, we see a good, strong relationship in the limited scope that we have. We have sites here, and we see this strong tendency for low flush of CO2, low yield without in-season nitrogen, compared to high flush of CO2 and much greater yield with nitrogen fertilizer. Almost astonishing that we could have such a strong relationship in field studies. We do have this one point here that is seems an outlier, but what we see is that there's with flush of CO2, we have a few sites that low yield, relative yield in season without nitrogen, come to that of the full nitrogen. And then we have this tendency for reaching the peak, where there's no yield effect due to nitrogen fertilizer with high levels of flush of CO2. We're putting the data together, which would be not advisable at this point, but I wanted to show you that actually there is, because I have limited data, I wanted to put them together. If we start putting the values together, it's that we could have such a strong relationship under different crops even, not only different fields of the same crop, but different fields of the same crop and of different crops. We're seeing this strong relationship. So I believe there's really good reason to be excited about using this as a soil testing tool. Utilized response to nitrogen, where we have available nitrogen on the x-axis and we have relative yield on the y-axis. We should receive no yield. With nitrogen, we receive plenty yield. It's up to define where that nitrogen comes from. So we get nitrogen coming from inorganic nitrogen, which we need to characterize surface soil inorganic nitrogen. We also characterize residual in the profile, because certainly there can be carryover from one year to the next, and there could be a significant amount of nitrate in the profile. We also need to look at the long-term stable organic nitrogen and the biologically active nitrogen. We want low nitrogen availability and hydrogen fertilizer response. We also characterize sites with high nitrogen availability and low nitrogen fertilizer response, because that's equally important, because we should be recommending to producers, if they have sufficient nitrogen, they don't need to add nitrogen. They need to utilize the nitrogen that's in their soil system already. And we should push this so that we have this enlarging biologically active nitrogen pool without causing significant nitrogen leakage. And the goal of the soil health is to increase the biologically active nitrogen pool without causing environmental damage. On the biologically active pool, because we believe that that's where we can actually add significant information to our current knowledge. But we need to characterize surface soil inorganic nitrogen as well. We can't work in isolation. We need to have a more complete package. And so these two are something that we can measure. It's more difficult to measure the residual nitrate in profile, but it should be done where there are reasons that there should be significant residual in the profile. And this long-term stable is actually something that we're actually looking at through our particulate organic nitrogen pool, as well as the total organic nitrogen. And perhaps the biologically active frac that we measure through the flush of CO2 is actually picking up a lot of the long-term stable nitrogen as well, because we know that it relates to long-term incubation. We need to use our soil resources wisely to be able to achieve sustainability. Soil quality has been the least understood, and therefore that's the reason for the recent emphasis on soil health to the biological component. And spreading of dried soil is related to these active portions of the soil through the soil microbial biomass carbon, nitrogen mineralization, and also to the more stable pool of the total organic carbon. As we developed a standardized procedure of the flush of CO2, and there is this commercial version available, and but the calibrations are needed to consider the variations that are there. Now, a commercial situation that it'll be easy. All you have to do is pay, and then you get your equipment readily available to you. But there, of course, is the cost associated with it. And to consider whether you have equipment or not, and certainly in our lab we have the equipment, so it's much easier, much cheaper for us to do it in the laboratory under the conditions we have. And frankly, I believe the procedure that we have is the most robust one that is out there. As a preseason test to predict nitrogen availability to crops is underway through repeating of our incubations in the laboratory, through the trials that Molly is currently conducting, and that Tangriani has conducted significant analyses as well, and others will be able to do this as well. And we're also targeting this field response, because ultimately this is what producers are going to be convinced of. They can be convinced of greenhouse growth trials, but they again, like most science, if they don't see it on their farm, they're not so convinced. So we're approaching this that if we can get reasonable data on field trials, we're going to make some headway on making this test available to them. So we demonstrated strong relationship with nitrogen mineralization under controlled laboratory conditions in similar soils with different management. The next data to suggest the strong relationship with plant nitrogen uptake under semi-controlled greenhouse conditions in a much larger, wider range of soils. So this is another strength of the approach we're using now in Molly's study is to use a number of different soils rather than just the same soil type with different management, looking at a number of different soils with different texture and different origin, different climatic conditions. There's a good relationship with the relative crop yield under field conditions in North Carolina and Virginia. And my is that we're going to be able to put to numbers in a more process-oriented way why we give credit to legumes such as alfalfa or cover crops. And this is an important step, rather than to give a category number of say that we can dock 100 kilograms of nitrogen per hectare an alfalfa crop. We should be able to use this test to actually put a more quantitative estimate from each individual soil. In the flush during 3 days is approximately 5 times the amount of nitrogen mineralized in 24 days. So I'm trying to say that we should, just because it's 5 times higher, but it means that there's greater sensitivity. Because the flush of CO2 is a one-way process as well, so we're not confused by the immobilization, although we have to be concerned about the relationships within immobilization. But I think that's a scientific question. But the soil test, it'll be less confusing if we actually just use the flush of CO2 because it's a one-way process. That it's a larger value means that actually we get a better estimate of the flush, and so we'll have a better route to say that we can actually say that you can dock 30 kilograms of nitrogen per hectare versus 50 kilograms of nitrogen per hectare. So a number of years is demonstrating that the flush of CO2 should be considered a robust soil testing for biological quality and nitrogen availability because of the following features. It's simple, and it's real. Contribute valuable information to define the boundaries of use. We don't know how this nitrogen immobilization will occur, and we need to define where the flex points are in blending fertilizer versus not. And we need to plant growth under various conditions. And the sustainability of soil and the environment will be enhanced by applying this knowledge because nitrogen is a significant source of pollution, but it's also a significant requirement for high productivity. And so it meets both of our extremes of how we're going to blend productivity with environmental quality. I have had significant collaborations, and I do have to mention many of them. We've got Virginia Department of Conservation and Recreation. We've got excellent collaborators there that have been willing to arrange for field studies. We've had studies on North Department of Agriculture research stations. We're the American Farmland Trust through kind of a joint venture. It's not an agreement yet, but we do have similar soils that we're working with together. We've got ARS people there in Beltsville. We've had significant collaboration with Dr. Carl Crozier, Deanna Osman, and Gail Overson at NDSU. And this process is greatly appreciated through from the USD National Institute of Food and Agriculture. Had cooperations with Penn State and Virginia Tech. And we've also had funding the NRCS through the Conservation Innovation Grants to help support this work. Willing to take questions from the audience in Brazil, and with my colleagues in Raleigh, I will speak with you afterwards. But essentially, it's a one-hour presentation, and the time is up. But if you have questions, please let me know. Thank you.
Sorry, we're having question for you.
Microfone ligado. Alguém está com o microfone ligado? Alguém está com o microfone ligado?
Hello?
Acho que agora você pode.
Very one that I have. I was wondering. I would like to know your about. Where do you think that we are at regional or national levels? Having the farmers using this kind of test to guide their nitrogen fertilization.
Okay, thanks for the question. You can hear me? Yes. The answer is, first of all, it's a great voice, and I'm glad to see you on the video there. It's a real pleasure. It was a great pleasure for you to be with us here in our laboratory. Currently, actually, it is being used by some soil testing laboratories in the United States. Brookside laboratories, they are using it because of the aggressive Rick Haney and Will Salvita are using. There are producers that are using the test. I feel that there's significant calibration that's needed, and that's the reason that we're doing the greenhouse studies. That's the reason we're doing the field studies. We need calibration. We can't. They have a strong opinion. I believe they're right. That I'm not willing to make recommendations without a strong database. That we're developing is important. Something that is going to take years to really fully. And to make it a regional or national recommendation, I think we are away. But there are recommendations through the Haney soil test that are being made. So people are using it. Thank you. Are there other? Okay. No, it's not working for lack of questions. I hope you recorded the session. We hope to post on some format so that you may be able to review it again if you so desire. Thank you all for attending. Now, thanks again. Thank you, Aníbal. Come on.